Whilst I'm waiting for my presentation to come up, good morning, ladies and gentlemen. It's a great privilege to be able to present this for a large number of people that are involved with this exercise over the last couple of years. I just want to make sure that you folk at the end can hear me okay. Is that coming through now loud and clear? Excellent. So, okay. I always get very relieved to see my slides come up rather than just standing here wondering what I'm going to say. Pardon?
Is this your presentation?
It is. Looks like it. Thank you. There's usually a picture of my mother on the second slide, so I can see that. So, if you can give that full screen, we can go. Okay, so I'm all right. So, right, good. So my job here this morning is to overview what was decided in these guidelines for the release of GM mosquitoes. So the exercise here is to go, what is the pathway of progress from the lab to actual final deployment in the field. Okay, so the background. So the exercise really was thinking about malaria control in Africa, and specifically the control of Anopheles gambiae. And really, the whole exercise was gauged around the idea of making sure that this technology would be safe and efficient before final field testing and deployment. So I'm pressing and it's not moving forward.
Let me try and close it and reopen it.
Okay. That's it. Beautiful now. Okay, great. Okay, so we considered two genetic methods of control. One is suppression, and the other one is population replacement. And I'll explain what that is in more detail in a minute. But first of all, this pathway of development didn't come from nowhere. It was based on WHO's work from the 2014 publication, which I can show here. This is a 4-phase procedure, and it starts with the lab studies. First of all, it then moves to a second phase of field testing in natural settings under strictly confined conditions, before moving up to a third phase, which is about entomological and epidemiological outcomes, and then finally deployment with continued surveillance. But the world has moved on very rapidly since 2014. There have been some major inventions, if you like, particularly with CRISPR-Cas9, which has transformed the scene. So this is one of the reasons to take a closer look. So the two basic mechanisms for the release of GM mosquitoes is either replacement or suppression. Now, if you look here at the top slide here, the black mosquitoes represent the wild population, the grey mosquitoes, the GM mosquitoes. And what you can see here is that progressively, with the drive mechanism, that over time, the genetic construct spreads throughout the whole population. Now, the important thing here is that you then have refractory mosquitoes to malaria mosquitoes, to malaria, hopefully, in the end. So you still have mosquitoes in the population. This is very different from population suppression, when the gene drive mechanism suppresses reproduction and causes a collapse in the mosquito numbers. So in essence, you may actually result in elimination of the mosquito on a very wide scale. So the spread of this gene drive requires consideration of a number of aspects. First of all, if you have escapees in the field, they may establish themselves in surrounding environments. And through sexual reproduction, that construct can spread. Importantly, it may spread so rapidly it can go across borders. So you start in country A and it has potential for moving across to country B. So that is very different from a number of other vector control interventions. Finally, you may have the position that the modification persists in the wild population. So what you're doing is very different from previous experiments. So this was, again, a reason that we need to revisit the 2014 recommendations by WHO. And I would say that in our discussions, we did work very closely with WHO, and we had members of WHO past and present. So here's my own personal piece, because I think a lot of people are concerned about, well, if we go for the population suppression and we eliminate Anopheles gambiae, does it matter? And here's my personal view, not the view of the team. I want you to remember that Anopheles gambiae is humankind's greatest killer. When I started working in malaria 30 years ago, there were 2 million children that were dying each year from malaria. Today, there's 400,000. Most of those are in Africa, and that's 400,000 too many. We have eliminated mosquitoes from Brazil and Egypt, Anopheles arabiensis. And don't forget that insecticides have been deployed on a massive scale, and we've killed trillions of Anopheles gambiae, and nobody has, as far as I'm concerned, has worried too much about that. Ecologically, it is rare in nature. You actually have to go and find and search for it in aquatic habitats, and you have to be a bit of a hunter. But it is beautifully attuned to finding us, so it occurs in large numbers inside the house. Now, I can only think of one example, there may well be others, of an animal which solely relies on Anopheles gambiae, and that's a small indoor spider in East Africa. I think that spider may go as development improves and the number of houses and house improvements, that spider will find it less favourable to exist. So overall, I think if you actually remove this mosquito, I think it would have little impact on the local ecosystems, but only for this mosquito species, because it is so beautifully adapted for people. So now here's the pathway that was developed. We start, of course, in the lab, and then we move down this pipeline from small-scale isolated releases to larger-scale, small-scale releases. Then finally, when we have confidence in what we're doing, in scaling up, we do large-scale open releases with epi outcomes. And then finally, massive deployment in an area for control. Now, part of the thinking of the group was enforced by people which had a wide range of experiences. And so these were not just molecular biologists and entomologists. They were people from the biocontrol environment where you're releasing new animals into new environments. And so we're learning from the methods and mechanisms that were established for doing that. And secondly, the release of genetically modified plants and the sort of obstacles and challenges that presented, as well as expertise from people that understood and designed clinical trials for vector control tools. So these are the highlights then. And so we're looking at the safety, efficacy, containment, monitoring, engagement, and regulatory aspects of this. So it's not entirely about thinking about the effect of the mosquito on malaria transmission. So this is providing evidence for a whole bunch of people, from researchers to regulators and funders as well, because this is a large-scale community that we need to think about. So in the laboratory studies, the recommendations are we should avoid premature release into the environment, and the sort of containment facilities that are required are arthropod containment level 2, which requires stringent physical control. And the candidates for field testing must show efficacy and fitness with the product claim. And in these early stages, it's really helpful to have modeling to support the outcomes because they aid with strategic thinking. But of course, all models are wrong, but some are useful. So they're not the real life situation. So partnerships and engagement, I think, are really critical from an early stage, which is why I think this publication is so important. If gene-derived products are developed from outside Africa, there must be strong partnership with African scientists and institutions from the beginning, in the early phases of development. And the partnership really must be in a spirit of co-ownership and co-development. This is a teamwork. And in-country institution must take a lead on regulatory and stakeholder engagement interactions. Now, this graphic shows you that just to sort of gives you a taste of the sort of involvement here. The research team is at the center of these bubbles, and this is both in-country and external research. But there are a lot of players involved in this, and probably many more than are shown here. So including the regulators, the ethics, NEPAD, which is the New African Partnership for African Development, ministries of health, NBA, that's not the basketball, that's the National Biodiversity Association, the media, and most importantly, the communities. So there needs to be an openness here. There needs to be an openness amongst the researchers themselves, and to avoid any rush to be the first, there needs to be sharing of where we are and with data sharing as well. This transparency is utterly important to earn the confidence of the public and reduce anxiety. And I actually think that if you want a better guiding light for this, the folk that have been doing the work on Wolbachia infected mosquitoes and the releases have been absolutely exemplary. They spend a considerable amount of their time in working with communities and a huge amount of their budget. And I think that really is important, absolutely vital. Okay, so it requires appropriate processes and platforms for broad sharing of information and make records of any releases, whether it's accidental or intended, available. Share best practices, publish baseline field data, and make public monitoring results. And this is something that we're asking the funders to think about as well. Important, those people that are going to support these developments. So where do we do it? Whereabouts in Africa? What are the things we need to think about? So first of all, we need a suitable biological environment. It must be targeting, don't make it too difficult. What's the major species there? Is it an Anopheles gambiae? Is it Sensus stricto, Kaluzi, whatever? And presumably, you want an area where malaria is not too low and not supersaturated, so somewhere in between. There must be no obvious social or political barriers, no widespread public or institutional opposition to similar technology. There must be support from the government. And there must be also the legal and regulatory framework in order to help progress these interventions. So it's important, it's not just doing the experiments, it's far bigger than that. And one needs to move forward progressively, carefully, and slowly. So the first situation is when you step out of the lab to go in to do small-scale releases. So it must be absolutely, well, as absolute as you can get, confident that your product is not going to do more harm to human health and the environment than wild-type mosquitoes of the same genetic background. Also, that resistance won't arise too rapidly to allow for positive health impact. And releases should not begin until funding is secured really down the pipeline, because once you start, then there is I think an ethical reason for carrying on, and that must be a long-term commitment, particularly the major consideration, of course, is the safety of the participants. Risk assessments are going to form an important part of the process, and these must be independent, particularly from that first step, moving from the lab to the field, and looking at both social and technical impacts. There's also a role here for self-limiting constructs. So that what we mean here is when you remove the drive mechanism, so the mosquito may only survive for a few generations or single generations. So this is not the final product, but it's a sort of halfway house to provide people with the confidence that this is safe. And it allows you to do all sorts of things to see how the mosquitoes survive in the field, but also it's an important training exercise. This is a massive learning process. Monitoring evaluation is key, and this needs to be thought through well in advance. And we need to develop new procedures for picking up escapees, for being sensitive to areas in which we're doing the releases. This must be agreed before the trial, and ideally it should be, again, done independently from those people that are doing the releases. And plans must be in place for remediation if there is pullback. So it's not just release and then close your eyes, it's following this through. So ecological surveillance is also required, and this must be done well in advance. So when one comes to do those first releases, what does it look like? Well, before you do releases, you've got to know really what's going on in the environment. So at least 2 years baseline entomological data and ecological data for at least 2 years is the recommendation. And whilst we're not being prescriptive, for some people are happy to use semi-field releases. So this is a semi-field site, and what is effectively it is a large greenhouse, where you can do releases under controlled conditions. But even then you need to be concerned for escapees or whether accidental or otherwise. But again, this could be useful for boosting support for the regulators and ethics committee that what you're doing is safe. So if you're going to do this sort of release, these sort of experiments must be done near sites where you're expecting to do the first field releases anyhow. But it also requires monitoring for escapees around the outside, just in case there are releases. So then we move on to the small-scale releases, looking at entomological outcomes primarily. So these experiments are done really to better understand the mosquitoes themselves and the drive mechanisms. So it's related to the rate of spread of the transgenic construct and the ability to suppress or replace vector populations. And it's important as well to monitor the construct stability to see whether it changes over time. So there's technologies that have got to be developed and scaled up in order to do that. It's important also to restrict environmental exposure. So these first releases need to be done either in island situations or in situations where they may be remote from, maybe surrounded by dry land and desert, but relatively isolated populations. It's important, again, the safety concerns is paramount to observe what's going on, and this may be be demanded, is likely to be demanded by regulators and ethics committees, and particularly looking at the interactions of the gene constructs with other mosquito species. So there needs to be, I think, when it comes to the environmental impact, there's a lot more thinking that needs to be done to try and to decide what are the key measurements that one needs to make, what are the organisms we're most concerned about, and to develop technologies for picking up those adverse interactions should they occur. We're not expecting them to occur. We start with the principle that this is safe before we do the releases, but we need to expect the unexpected. any evidence for adverse impact would be a no-go stop, so for further testing. It may be important, I think it will be important, to do multiple releases in different sort of environments, because it may work well in one area and not the other for various ecological reasons. It may be too hot or whatever. Large-scale releases, then here we're looking at the impact on disease transmission and spatial dispersal, and particularly what we're interested here is not the effect on the mosquito, but is it reducing malaria in the local population, infection and incidence. And yeah, need to consider other malaria vectors. We need to monitor the effect on population structure and on non-target organisms. So that's fine. And then we go to implementation, and this requires decision-making to go from regional deployment to regional deployment. So a very important aspect of these releases is regional conversations, avoiding surprises, and because it's a wider issue here. If one goes to scale up, that decision is based on the efficacy of the construct, the safety, the perceived benefit, acceptability, and the cost. This should be not considered as a standalone intervention. This is integrated vector management. It's additional to the other vector control tools that we're deploying across sub-Saharan Africa. It's not a cheap fix. It's an additional tool. It's very important to WHO to play a leading role in this, and I'm sure that they will do, because it requires conversations across countries, across regions. It requires long-term planning for the future. Business models need to be developed, engagement with governments, quality control and standards need to be thought through. And after implementation, surveillance doesn't stop, it carries on. And again, WHO here has a key role because of the scale of these operations. And it's going to require considerable amount of injection of capital to continue to do the surveillance to make sure that it's working. And that includes not just watching what's going on with the construct and seeing whether there's resistance developing, but potential adverse effects on the ecosystem. And as with all interventions, resistance may well occur with all interventions. So what are the resources? So what do we need to make this work? More sensitive monitoring tools for escapees, standards and best practices, information and data sharing, support for risk assessment, and really thinking through what is required. Support for effective stakeholder and community engagement, opportunities to build capacity in regulation of modified insects, regional harmonization, quality standards for manufacturing of these living products, and mechanisms for oversight and management post-implementation, and the development of assays for product performance failure and systems for reporting performance failure. Finally, the coordination here is key. From the perspective of product development, inappropriately conducted field trials have the potential to negatively impact the future success of other gene-derived products, which will undermine the community, stakeholder and public confidence, and to contaminate the regulatory and funding environment. Here is a plea for people to go slowly, carefully, because what you do will affect the series of interventions, which has potential to make a great difference to malaria across sub-Saharan Africa. We're all in this together. We don't need the woodpecker picking holes in the boat to make it sink prematurely. Thank you for listening.